Bismillahirrahmanirrahim. Continuing the previous lecture, which was about the applications of bioinformatics. So we have stopped. We have discontinued our lecture on personalized medicines. Now we will be moving towards the preventive medicine, as this is a part of that medicinal approach and applications of bioinformatics in the field of medicine. In preventive medicines, what we do, we usually check the susceptibility of a person to a disease. Like for example, with specific details of genetic mechanisms of the disease being revealed, the development of diagnostic tests to measure a person's susceptibility to different diseases may become distinct reality. Preventive actions such as change of lifestyle, having treatment to the earliest possible stages when they are more likely to be successful, could be a result in huge advances in struggle to conquer a disease, like changes in different lifestyle. So usually what we do in preventive medicine, we provide different types of, you can say, those medicines which are, like for example, you can take an example of probiotics, which helps in improving the growth of normal flora, and different types of those medicines which can only boost your immune system, that can help you in fighting against that specific disease. So this is the detail about the preventive medicine. What is gene therapy? Basically, gene therapy is a molecular level tool, or you can say a treatment on molecular level. Gene therapy is the approach used to treat, cure, or even prevent disease by changing the expression of a person's gene. So what we usually do in gene therapy, that for example, if you have find out a gene which is having a problem, or if for example you find out a gene which is having a missing code, or missing codons, 1, 2 or 3 nucleotides are missing, or for example 1, 2 or 3 nucleotides are additionally added, additional. So what we can do, that in gene therapy we study that specific gene which is causing the problem in your in the person's genome, and then we remove that. So it is a type of therapy you can add, you can remove, and you can suppress a gene as well. So it in not the too distant future, the potential for using genes themselves to be treated, to treat themselves to disease, may become a reality, and it is now has become a reality, because in different European countries and America is following the techniques of gene therapy. Microbial genome applications, in this microbial genome application, we are now going to study that you can see that microbes are present over everywhere over the world, in every part of the world, like for example in air, in soil, in water, and even in hot streams as well, because they have a very ubiquitous nature. Then they are present in a very harsh environment as well. So what we do, we can take the help of these microbial organisms, like for example getting a gene that is present in in a bacteria which is growing in harsh environment like heat, or you can say in extremely cold environment like -21 or -121 degrees centigrade, etc. So we take that gene which is producing a type of protein that makes it able for the bacteria to grow. So we can take an example of, we can take a good help from that specific, you can say bacteria. तो ये जो है माइक्रोबियल जीनोम एप्लीकेशंस जो है, इनकी एप्लीकेशंस भी हम लोग जो है यूज़ कर सकते हैं Can begin to understand these microbes at a very fundamental level and isolate the genes that give them their unique abilities to survive under extreme conditions. माइक्रोबियल जीनोम एप्लीकेशन आपके साथ कहाँ कहाँ पर हेल्प कर सकता है? इट इंक्लूड्स द फॉलोइंग: वेस्ट क्लीन अप, climate change studies, alternative energy resources. You can get methane producing bacteria. You can produce methane gas and other energy resources as well. In biotechnology, antibiotic resisting, forensic analysis of microbes, bioweapons. In forensic analysis, usually after when in forensic, they sometimes check the availability of microbe, because sometimes microbe in dead body are changed, like, and different types of bacteria and different types of microbes are produced in the dead body. So they can also take help from that microbes as well, कि कभी कभी जो है डेड बॉडी में माइक्रोब्स की प्रोडक्शन होती है, तो वो उसको स्टडी करके बताते हैं आपको कि ये डेड बॉडी, ये जो इस पर्सन की डेथ कब हुई है. तो यूजुअली हम इसको भी यूटिलाइज करते हैं. बायो वेपन क्रिएशन, एवोल्यूशनरी स्टडीज एटसेट्रा. वेस्ट क्लीन अप, जिस तरह मैंने आपको बता दिया है कि माइक्रोबियल जीनोम प्रोग्राम साइंटिस्ट आर डिटरमाइनिंग द डीएनए सीक्वेंस और जीनोम ऑफ क्रेसेंटस, द ऑर्गेनिज्म रिस्पॉन्सिबल फॉर सीवेज ट्रीटमेंट. तो ये इनका जो है एक मिशन है. क्लाइमेट चेंज स्टडीज जो है, वन ऑफ द मैटर ऑफ डूइंग इज टू स्टडी द जीनोम्स ऑफ माइक्रोब्स that use carbon dioxide as their sole carbon source. तो हम क्या करते हैं कि हम माइक्रोब्स को यूटिलाइज करते हैं ताकि इनको हम स्टडी करें, और फिर जो है ये माइक्रोब्स जो हैं कार्बन डाइऑक्साइड को अपने लिए एज अ सोर्स ऑफ कार्बन यूज करते हैं, कार्बन डाइऑक्साइड को. तो हम जो है क्लाइमेट चेंजेस इसकी वजह से ला सकते हैं. इंक्रीजिंग लेवल ऑफ कार्बन डाइऑक्साइड इमिशन मेनली थ्रू the expanding use of fossil fuels for energy and thought to contribute global climate change. Alternative energy resources, scientists are studying the genome of microbe Chlorobium tapetum, which has an unusual capacity for generating energy from light. ये देखें, ये जो एक इम्पोर्टेंट जो है एक माइक्रोब है जो कि आपके साथ इन लाइट के साथ जो है जनरेटिंग एनर्जी, लाइट को देख के या लाइट को फील करके जो है आपके साथ एनर्जी को प्रोड्यूस करता है. ये एक बहुत इम्पोर्टेंट टेक्नोलॉजी है. ये हम इसको बायोइन्फॉर्मेटिक्स के ज़रिए इसकी प्रोटीन स्टडी करते हैं. बायोटेक्नोलॉजी है. सो वी विल लीव दिस these slides.